All right, so in this video, I'm going to go over chapter 8, just the part on enzymes first. Um, so first and foremost, you just need to understand the keys in terms of what enzymes are going to be used for. But before we do that, let's get into a couple of terms that you want to remember. What is metabolism, and what's a metabolic pathway? So when you think of metabolism, that's talking about the chemical reactions in a body in the sense that don't think of it as like someone has a fast or slow metabolism. Anytime we have what's considered a metabolic pathway is where you're typically going to be starting with one molecule and you're going to be ending with a final product. But if you look at this picture or this diagram down here, it's showing you that it takes multiple reactions to get to that final end product, where going from A to B takes enzyme 1, going from B to C takes enzyme 2, and going from C to D requires another specific enzyme. So going from A to D, there's 3 steps to get to that final product of D. So that's what we're going to call a metabolic pathway. Now, couple of common AP test questions would be something along the lines like, what's going to happen if you increase the concentration of A, but let's say something happens to where enzyme 2 is no longer available. What is going to happen to your product of C and your product of D? Well, if this whole enzyme is not functioning for some reason, whether it's mutated or denatured, then reaction 2 doesn't take place. So if you increase the amount of A, enzyme 1 is still good to go. You can still increase your concentration of B, but your concentration of B is going to stay high because enzyme 2 doesn't function. So then the whole rest of the pathway gets shut down. So that's just a really common type of question you might see under test. Now, I also want to remind you of these 2 words, catabolic and anabolic. Catabolic means you're actually going to be breaking down, where anabolic means you're going to be building something. So think of things like cell respiration. What are you really doing? You're going to essentially be you're going to break down glucose. So you want to understand what that is referring to. So you're going to break down glucose in respiration. Anabolic essentially means you're building something. So anabolic pathway would be something, for example, protein synthesis. You're building those proteins. Or you could also just do the counterpoints, respiration, which is going to be photosynthesis. So anabolic means to build, catabolic means to break down. So know those 2 terms. And typically in biology systems, we're going to be releasing energy when we're actually going to be breaking something down. And that's because energy is released when you actually are going to be breaking apart a lot of this bonds. And a couple of things it has to deal with the keys of what is your reactivity of your products. Don't get too caught up in this. Those that are really, really good at chemistry, I will say this, that typically you get confused when you think about if you break bonds, it actually requires energy to do so. We're talking about a different mechanism here. So just understand that when you're breaking something down, typically you're releasing energy. Anabolic, you're consuming energy to actually build that up. So we're not specifically just talking about the bonds. So just know catabolic, breaking something down, anabolic means to build it. So when you think of energy, our biggest focus really for this one is going to be getting into a little bit of Gibbs free energy. We don't have to go too crazy. You just have to know that if it's an exergonic process, energy is released, something like cell respiration. If your delta G is negative, it's spontaneous. That means it is going to proceed in a biological system. If it is a positive delta G, it's endergonic. It requires energy in the form of things like ATP or sunlight, for example, in photosynthesis. But one big thing that we got to get into is what's called energy coupling. So let's just take a look here. If this is one step and this is another step and this is the overall equation, let's just look here. If you're trying to take glutamine, putting it on ammonia to make glutamic acid, that's actually going to take a good amount of energy, right? So this is showing you that if you're trying to put that glutamic acid on ammonia to actually make your end product of glutamate, your delta G value is a positive 3.4 kilocalories per mole. So that just basically means that if I put these 2 next to each other, it's going to take some energy. It's not going to naturally want to do that. But if I couple it, right, the key word is coupled. If I put ATP in the presence of that glutamic acid, ATP donates a phosphate. And that one phosphate makes this thing super unstable. And then that's going to allow that ammonia to come and bind. So basically, when you think about it from this standpoint, When you look at this phosphorylated intermediate, this makes it unstable. And any time you actually break down ATP, that is an exergonic reaction. So basically, when you look here, ATP to ADP, that is a really exergonic process. So basically, if you look at that delta G value, it's negative 7.3. If you combine it with ATP, the net or the overall spontaneity of this whole process is negative. So it makes it favorable. And the key player on this is ATP. So let's kind of just do a brief explanation as to why. If you look at ATP, it's in the nucleotide, right? So here's your base, here's your sugar, and here's your phosphate. So I just want you guys to remember that ATP is most definitely still a nucleotide in terms of the macromolecule. Base, the sugar, the phosphate. But these phosphates right here on the end, man, it's very, very unstable. So by giving up that phosphate, it typically wants to get rid of that. So it's relatively unstable. By getting rid of that phosphate, it makes it a lot more stable. So typically it wants to get rid of that. So in terms of that, this is kind of a nice little overview where it says ATP, if you hydrolyze it and you make ADP, that's going to be exergonic. When you put a phosphate back on, that actually takes energy to make some ATP. So it's not a real value that you have to know, but it's kind of a nice overview of adding in that phosphate to ADP to make ATP is actually endergonic. So that's what chapters 9 and 10 are all about in terms of how to make this thing favorable. So anyway, the big focus for this one is really getting into enzymes. So before we get into ATP, I just wanted to kind of recap what are enzymes. First and foremost, you need to know that enzymes are proteins. I can't emphasize that enough, how important that is to remember that enzymes are proteins. And another thing too that is massively important is that each enzyme is specific to the substrates. So basically what this means is that if I had sucrose, so sucrose is a disaccharide, and I actually wanted to break it up, right? Well, if I added water, it's not necessarily going to innately break it down, but if I have an enzyme sucrase, so I'll draw my little Pac-Man looking enzyme here. Sucrose and water can fit perfectly in this enzyme called sucrase, okay? So in the presence of that enzyme, it's going to make this process function a lot quicker. So enzymes are going to be considered catalytic proteins. And it's going to help hydrolyze it in this case. So basically, if I took sucrase, sucrase isn't going to work on anything else but sucrose. And just to remember, all enzymes are going to end in ACE. It's an easy way to identify if it's talking about an enzyme or not. So moving forward, really the key thing about enzymes and what they do, they're going to help lower the activation, that activation energy. It's the energy required to get a reaction going. They're going to help lower that activation energy. And let's kind of help explain why. So I'm going to move over to this simulation from Mastering Biology. So if you look here, how do enzymes actually work? So this is showing you that if you have a lot of energy and then less energy at the end, that's an exergonic process. But if you look here, here's your reactants. A and B and C and D are your reactants, and it's going to take that enzyme. The enzyme is basically going to require, or I shouldn't say require, it's going to provide the perfect micro climate for this reaction to actually proceed. So if you let this play out, enzymes are going to serve as that biological catalyst. They increase the rate because they actually lower that activation energy. They make this reaction easier to proceed. So, and it has to deal with the actual shape of these molecules. So don't get so fixated on what these reactants are, or really what your product is. This is just talking about all enzymes do, and you can see that A and B fit perfectly here, C and D fit perfectly here. This region of the enzyme is called the active site. So it just shows you that this protein, this enzyme, is specific to these molecules, A, B, and C and D. So it speeds it up by making this reaction easier to proceed. So enzymes are very, very selective, as it's stating here. It allows it to bind specifically here, and it makes this reaction proceed very, very easily in terms of providing that perfect microenvironment. So if you let this play out, we can see what's going to go on here is that A is going to bond with C, B is going to bond with D. And then at the end of this, B and D have less energy than it did initially when B was bonded to A. So this is just kind of showing you an overview. Having this enzyme made that reaction proceed a lot easier. So understand that concept. So in terms of this, this is talking about thousands of different enzymes maybe required to carry out all these metabolic processes. So again, watch, B is going to bond with D, A is going to bond with C. These are your end products. A and C has less energy than when A was bonded with B earlier. So this is showing you it's an example of an exergonic reaction. So let's get back to the notes. And now you can kind of see this, this red line is showing you that when an enzyme is present, it makes it easier for that reaction to proceed, okay? Now the vocab here is really, really critical. So let's kind of take a second and emphasize this. Substrates are basically what you're starting off with, and then obviously your products are going to be what's going to be at the end when this whole thing is complete. So I like this because it kind of shows you this nice cyclical process. Your substrates bind to the enzyme. Where they bind, this little area is called the active site. The chemistry magic all takes place within the enzyme. Your products are spit out, and then here's your enzyme at the end. Notice that the enzymes don't get used up. They're there at the beginning. They're there at the end, okay? Now, one key thing that a lot of students misinterpret is, well, they know that the substrates fit to the active site, but why does that matter? Well, if you look here, it tells you it acts as a template. That means that it makes it perfect for these two things to actually bond. It helps them orient themselves correctly to actually make a bond or break a bond, depending on what it's doing. It can actually help stress the substrates, and actually in terms of if it's trying to break a bond, it provides a favorable microenvironment. So I don't want to use the word distracted, but if you want to think of it in terms of there's no other ions around, no pH changes, it's going to help provide that perfect environment for these things to actually do the reaction that is needed to take place. And then they participate directly in that catalytic reaction. So in terms of that, this is kind of a nice little overview here. So if you look here, here's your substrate binding perfectly to that enzyme. This substrate can't fit. I really, really love this one, that if you add these protons, you're lowering the pH, it changes the shape. This protein is actually denatured. So I really, really love the visuals here. So I'll kind of go back and let this replay out. If you look up here at the top right again, you're gonna see if you add these protons, you're actually lowering the pH. By changing the pH, you're changing the shape. This protein is now denatured. Substrates don't fit, okay? Now again, this is just kind of really the key in terms of what do they do, or why do enzymes actually work on lowering the activation energy? Just kind of a reiteration of what I just said. So basically this is just talking about really that first point. A and B have to be lined up next to each other perfectly for this to take place, to actually form that bond. So when it says orient them correctly, it's actually providing that environment for them to actually be facing correctly. And in terms of that, that's kind of the key one that I think a lot of people misinterpret. But just remember this, that each enzyme is specific to the substrates, okay, and you get your product. And here's just kind of another visual. When this substrate goes into the enzyme, this thing right here is your active site. So I'll just abbreviate that as active site. Notice that the protein or the enzyme actually changes shape. This process right here, it's a really fancy word, but when your substrate is actually in the enzyme, we get to call that an enzyme substrate complex. So basically know that the protein does change shape during the reaction, and then it basically, as a product is released, the enzyme changes back to its original shape. So one big thing that the AP test loves to trick you guys on is how do these conditions change. And so the two big most common ones are going to be temperature and pH. And the big thing about this is that if those change too much, you're actually going to denature your protein. So the protein no doubt gets denatured in the sense that it's no longer functioning. And that makes sense because when you do this, if you change the pH, it gets too acidic or it gets too basic, the protein is going to start to change shape. Same thing with the temperature. They're going to actually denature. Okay. Now concentration and cofactors, basically this is going to help change the rate. Okay, and it depends on concentration. We can talk about the concentration of two things: the concentration of the enzyme, and you should also know about the concentration of the substrate. So I'll do a graph to show you the difference between each one. But when you think about concentration, if you added more enzymes, if you added more substrates, you want to be able to understand how does that change the rate, okay? Cofactors are going to be things that are going to actually be added to the enzyme to turn them on or turn them off. So just kind of a nice little overview of what's happening here. If you added in your enzyme, it's going to lower the activation energy, so it's going to make it easier to proceed. Notice the overall delta G, or the energy released in this case, is not affected. It just makes it easier to proceed. Okay. Now this is taken from your POGL. This one shows you a couple of different examples here. So if you have triacylglycerides, which are fats, you want to break that down into glycerol and fatty acids. It takes the enzyme lipase, so that is your enzyme right there. And I have it to where lipase is in this type of category. Lipase, you would look at a graph like this, and it tells you the rate of the reaction is the highest at a pH of 8. That's the most optimal pH. Now, if you look at large polypeptides, which are, again, these big proteins, it takes the enzyme pepsin. But if you look here, pepsin looks to be the most optimal at a pH of around 2. So it just shows you that some enzymes can violate that rule. Really, the only one that we'll talk about and that you would really probably ever see is pepsin, because pepsin works in your stomach, so it actually works best at a pH of 2. But if I actually took pepsin, put it in a pH of 8, it wouldn't work, and vice versa. If I took lipase and put it in a pH of 2, it wouldn't work. So let's kind of talk about enzyme inhibition and how do we turn on and turn things off. So it's just as important to turn on an enzyme as it is to turn it off. So let's talk about inhibition. So a competitive inhibitor versus a non-competitive inhibitor, both of them actually are going to stop the enzyme from functioning. So basically what I'm trying to emphasize here is that what is the difference between a competitive and a non-competitive inhibitor? So if you look right here at this enzyme, if you added a competitive inhibitor, notice that this competitive inhibitor actually binds to the active site. So it's actually competing with the substrate. The name itself almost implies what it's doing. So that way you have something blocking the substrate, so the substrate can't bind and the substrate actually is going to leave. So in terms of what's a competitive inhibitor, think of it as this inhibitor competes with the active site. Now, if you look here, you have another molecule that's bound to the enzyme, but it changes the active site's shape. So even though there's nothing blocking the active site's shape has changed, your substrate cannot bind, and then it essentially just has to leave, or it can't do anything. So by adding this molecule, it changed the shape of that protein. Now it makes it unfunctioning. So just be careful with your wording here. Don't necessarily say that this is denatured, because you're not changing the temperature of pH. You just have something changing the shape of the protein because it's bonded to the enzyme. So don't call this denaturing. Now let's talk about allosteric regulation. So how do you turn on or off an enzyme? So like I mentioned earlier, if you have something that's binding not to the active site, but somewhere else, we're going to say that it's binding allosterically. So if you look here, the active site won't allow this substrate to bind, but in the presence of that allosteric molecule, you cause the active site shape to change just a little bit, and now this molecule can bind, and essentially now it is turned on, okay? Now, if you look at the contrary on the other side, the active site shape is typically on, your substrate can come through and do its magic, but binding this molecule allosterically, it actually turns it off. So just note the difference between an activator and an inhibitor. Activator, as the name implies, it's something that's needed to turn on an enzyme, and an inhibitor is really, really important too. In fact, most of the time on your AP test, they like to test you on inhibitors more so than anything else, because by adding an inhibitor, a lot of people think that that's a bad thing. It's not, because this is eventually going to go and make your product normally. So it's good to make your product, but if you make too much of it, sometimes your product can actually bind allosterically to prevent overproduction of it. So these are naturally occurring. It's good to turn them off just as good as it is to turn these things on. So know the difference between how these things get regulated. So this last one is on feedback inhibition. So basically this is showing you that if you have this initial molecule, it's this nice metabolic pathway, multiple steps here, you make your end product. But if your concentration of your end product starts to increase, your cell doesn't want to overproduce this end product. And so keep in mind, as I told you guys a bunch of times in class, typically you have, you know, hundreds of each one of these enzymes. So think of almost multiply these in your head by like 100, if not 1,000, maybe even 10,000. But your end product, it's not, don't read this as just like one essential substrate is going to make one product and then you stop the whole thing. Typically, you're going to have hundreds of these, okay? Like I said, if not thousands. So it makes sense when you think of it from this standpoint, that if you made thousands or 10,000s of these products, they're going to bind to this enzyme allosterically, right? And it turns it off. So that way it lets the cell know that it prevents wasting. So, and as this concentration starts to decline, this gets cleaved off and then the whole process gets turned back on. So that's the simple and really classic way of explaining feedback mechanism, right? So the thing I did a bunch in class, if I had A makes B, makes C, makes D. If I had enzyme 1, 2, and 3, typically D can bind allosterically to enzyme 1 if the concentration gets too great. So moving forward, I think that's kind of the biggest thing. But let me just draw this graph out for you guys really quick. Let me try my best here, talking about the rate. So let me adjust my pen here. Well, we'll get this across here, okay? So let's say if I had a graph that looks like this, and this is time, and this is your product concentration. So we got a rate of reaction, okay? So yeah, let me change the color here real quick. All right, so let's look right here. If I had a line that looks like this, okay, so let's talk about it. If I talked about what is the rate here, remember it's your delta Y over delta T. So let's say time, this is in minutes. In terms of this, the rate is actually a positive value because you're actually making more product. Now what happens here is that your rate actually equals 0. And you need to understand why. Well, if I started out, if A plus B makes AB, right? If I had 10A plus 10B, I could make 10 of those AB molecules, or I'm just saying I'm forming a bond, right? So keep in mind, right here, all of your product is formed according to your substrate, right? So that's what you need to understand here, that if I had 10A and 10B, I can make 10 of my final product of AB, A bonded to B, okay? So why does it level off here? Well, you've typically used up all of your substrate. Now, if I added in another line that is going to look something like this, I'll do it in blue. If I added in a line that looks like this, all right, what did I change here? Well, I actually increased enzymes, okay? And the reason why I know that I increased enzymes here is because I didn't make more product, but I did make more product faster. So that means, for example, if I just, I'll actually put this value of 10. So product concentration of A, B. I made my max amount of product, but I just made that product faster. So I essentially added in more enzymes. Now, there's a couple of other scenarios that you want to make sure that you're feeling comfortable with. What if I, well, what if I had a line that looks something like this? Is going to get kind of messy, but right here. Well, a couple of things here, that if this purple line was used, well, what did I do? I actually increased substrate. Why do I know that? Well, again, I actually made more product. So if I call the black line my control group, so I would say that is my control. This is where I actually increase substrate. This is where I increased enzymes. Hopefully that's making more sense. Now, typically on your AP test, you would also have to draw out stuff like, well, what happens if I had a line that looked like this, or essentially is 0. Well, you need to make an assumption that you decrease the pH or you change the temperature, or you can mention something that there's no substrate or no enzymes. All of those are correct, okay? So just keep that in mind, that this is a graph that it's really imperative that you kind of have a basic understanding of how it works. And in terms of that, I think that's kind of one of the bigger things that I wanted to mention here. So yeah, we'll go ahead and leave it there for enzyme and a nice little recap on what these things are.